Oglala Meter Ranch, where we will be discussing today the structure and function of genes. I am Adri.
I'm Serena, and today we have a very special guest with us. He is a Ebola virus specialist, and he's going to be telling us a little bit about Ebola itself and a new groundbreaking paper that addresses Ebola virus gene structure and function.
Thank you, Serena. Hello, I'm Spencer, said Ebola virus expert. So, a little bit of background regarding Ebola virus. It is a part of the Filoviridae family and is a class 5 virus, meaning that it is a single-stranded negative sense genome. And it is highly pathogenic with currently no real treatments or vaccines present in order to help effectively treat it. Some distinct features of Ebola viruses that the paper is specifically concerned with are regarding its cis-acting regulatory elements, and specifically some directly overlapping transcription sites in signals, and then additionally there are exceptionally long intergenic regions present within its genome. And so within the paper, titled Analysis of the Highly Diverse Gene Borders in Ebola Virus Reveals a Distinct Mechanism of Transcriptional Regulation by Brauberger et al., they sort of aimed to assess these aspects of the Ebola virus genome in a little more detail.
Thank you so much, Spencer. So now we're going to head over to one of our field reporters, Jason, and he's going to be interviewing some people that are going to tell us a little bit more about the experiments done in this paper.
Thanks, Serena. I'm here now with Lily, and we're out here to discuss a little more about the paper. So, Lily, why don't you share with me a little bit about what questions the authors were trying to answer with this paper?
Right, Jason. Well, probably the main question that the authors were trying to address was how the diverse and unique gene borders of the Ebola virus genome would affect transcription of the virus, and that would help them better understand the virus in general to be able to address creating a vaccine or different treatments for it.
Awesome, thank you so much. So what were the first steps you had in getting the process started and finding out some results from this?
Right, so what needed to be done first was kind of just map out the genome in general. So if you look on the figure that's going to be presented on the slide, you can see that the genome has about 7 different genes, and those are separated by different gene borders, whether that be long IRs or short IRs, or also gene overlaps. And
do you mind if I stop you right there? What's an IR for those that may not know?
Right, so an IR is an intergenic region, and those are short sequences of nucleotides that are between different genes.
Okay.
And the authors were specifically looking at the long intergenic region, which is about 144 nucleotides long.
Oh, wow.
And then they used a northern blot to basically map out the different mRNAs that are transcribed from those, and to determine which mRNAs are transcribed more or less.
Awesome, thank you. Well, I'm now here with Tatum and our friend Kitty, don't know her name, and we're here to dive even deeper into the paper. So, Tatum, I was just with Lily, and we discussed actually analyzing the gene itself and identifying the various regions of it. So what were your next steps after that?
Right, so you've identified the different genes in the genome and identified these intergenic regions and overlapping sequences between the genes. And our next step was to see what that downstream effect of these intergenic regions are on transcriptional activity.
So how exactly did you guys go about doing that?
Yeah, so to look at the effect of these intergenic sequences on mRNA expression, we designed bicistronic mini-genomes, which are sequences of DNA that only contain 2 of the genes with an intergenic region, which acts to isolate that region from the rest of the DNA, or from the rest of any impacts that the other areas of the genome would have on expression.
So tell me about the results that you guys found.
Yeah, so we took these bicistronic segments and transfected them into living cells with an Ebola infection. And then we looked at the resulting mRNA that was transcribed from that. and we found that with our bicistronic mini genomes that had a long intergenic sequence, there was more transcription of that first gene compared to the second gene, meaning that with the long sequence, the RNA polymerase had more time to dissociate, more opportunity to dissociate, resulting in less transcription of the gene.
Wow. Thank you so much, Tatum. That was awesome.
Thank you.
So we're back here with Spencer and a special guest, Mickey, right behind us. And we're here to further dissect this extremely fascinating paper. Spencer?
Yeah. So next we wanted to sort of look at how exactly the effects of the IR and specifically the length and or sequence of this intergenic region sort of impacted the resulting downstream gene expression. And what we first wanted to look at was we first totally removed the intergenic region to see sort of the downstream effects of that. And as you can see from above, what we found was that the mRNA2 levels were significantly lowered in the group without that intergenic region, however, still present, indicating that this intergenic region is not necessarily required for that mRNA2 to be expressed.
Very interesting. So did you guys try anything more besides removing the intergenic region? Did you perhaps try extending it or shrinking it?
That's exactly what we did, Jason. Thank you for bringing that up. So next we tried extending that intergenic region, that long intergenic region in length to see if that had any downstream effects on the gene expression. And similarly to kind of what we found with that removal group, we found that there was a lower gene expression of that mRNA2 within that further extended group, which sort of just confirms that that intergenic region length is sort of very important to get that mRNA2 expression.
Wow, very interesting.
So then building off of that, we decided to then sort of truncate gradually the length of that long IR to sort of see how that impacted the resulting downstream gene expression. And so as we can see through this figure here, it was shown that as we slowly truncated that the length of that intergenic region, it was found that as you bring that shorter and shorter to a certain threshold, there's a region where it just get significantly less expression of that mRNA2 present within these bices, which sort of indicates that, again, like this length of the intergenic region has certainly a big impact on sort of the downstream effects of the expression within that genome.
Wow, Spencer, that was extremely enlightening, and I really enjoyed you sharing that with us today. I think our viewers are going to love this. And I'm Jason, and back to Adrian in the studio. Well, wasn't that just fantastic? So there's a lot we learned today about the Ebola virus that really wasn't known before we heard that from your paper. So Spencer, would you mind actually trying to wrap that up for us as a group into just a few short sentences? So our viewers have a better understanding of really what we dug into over there.
Yeah, absolutely. Thank you so much, Jason. So the big kind of overarching thing that was found from this research was that sort of these long intergenic regions are involved with sort of the downstream regulation of this gene expression within the Ebola virus.
So it seems that the intergenic region is playing a big role in that downstream regulation in its length, but are you sure that it's not just the sequence too?
Absolutely, yes. We can confirm with 100% certainty that it is sequence independent, meaning that it is not associated with the sequence at all, but rather the actual length of the intergenic region itself that is driving this regulation and the gene expression. And it was additionally found also that sort of these gene end signals that are present can help regulate sort of the read-through of this mRNA and are involved with the RNA polymerase dissociation and reinitiation of the Ebola virus.
Wow. That truly is fascinating, Spencer. Thank you so much for your time and coming here and sharing your knowledge with us, and not only us, but our viewers as well. They really want to hear this type of content, and they are asking for this, and we are so glad we were able to bring you onto our set and have you share this story. That was truly amazing. Thank you so much.
Yeah, thank you, Spencer, and thank you again for joining us at Lalameda Ranch.
Until next time.